All right, so let's go over this free response question. And I've mentioned this many times before, but you're going to get thrown things you've never seen before that look kind of crazy, but when you kind of get all the crazy nonsense stuff out, the questions kind of boil down to stuff that you already know or are very close to knowing. So it's going to be a lot of kind of extraneous information that might not be useful for the question. But so we have this study, right, looking at yeast, and they've got an experimental strain that produced a modified polymerase. That is because of one amino acid difference. They determine the maximum elongation rate during transcription with and without the modified polymerase enzyme, figure 1. They did a second experiment where they had this compound, amatitin, which is commonly found in toxic mushrooms, don't need to know that, is a polymerase inhibitor. And then the amatitin binds to an active site, polymerase active site, inhibits transcription. So the second experiment, they treated both types of yeast with amatitin and recorded the rate. And they've got error bars, and I explain error bars in the video you're hopefully watching today. So A, describe 3 structural components of a nucleotide monomer. Alright, that's the first part, and this is just basic knowledge of the macromolecules. Alright, we've got the ribose, the phosphate, and the nitrogenous base. So those are the 3 parts of the nucleotide. And then the role of polymerase during transcription. Transcription being DNA to RNA, so it's going to be RNA polymerase. And its role is to make RNA from the DNA template and to join those RNA nucleotides together. So hopefully everybody got A. Pretty easy. B, identify the dependent variable in experiment. And then identify a control group missing from the second experiment. Justify the need for this control group in the second experiment. Alright, so the dependent variable, again, referencing the video that hopefully you watched already today. The dependent is the same as responding variable. So you'll see both these two terms. So to define that, all you gotta do is look on the y-axis, right? Dependent and responding variable are on the y-axis. In this case, it's the maximum elongation rate. And you can put the units there, nucleotides per second. Identify a missing control group. Identify a control group missing from the second experiment. Justify the need for this control group. All right, control groups are there to kind of show you if there's any difference between what you're doing and what the normal situation is. All right, so the control group would be for the second experiment. Right, we don't know what effect there would be on the first experiment ones, right? So they just took the second experiment, they're just adding this compound from the mushrooms, amatitin. Right, so a control to see if there's a difference between if amatitin actually affects either one, you would need to compare it to the experimental strain here without amatitin, or the wild strain here without amatitin, or even, frankly, it'd be better to have both in there. To see, again, for the second experiment, you're seeing the effect of amatitin, but you don't have any good baseline to judge it, how drastic the effect is based on the other results, right? Maybe there's something in the conditions, again, they should be under the same conditions, that affected this. We don't know. So you need a control, and that control will be something from the first experiment without amatitin. Where am I? Okay, so we justified it. Oh, look at that. I had no idea I could zoom in. So we justify that because we need to know, right? The amount of this control we don't know. Again, the effect with amatitin. Without amatitin, we need to see that there. Alright, so where am I? C, describe the effect of amatitin on the maximum elongation rate for the wild type and modified polymerases. Determine the ratio of the average maximum elongation rate for the modified polymerase compared to the wild strain polymerase in figure 1. All right, so the effect of amytin on the maximum elongation rate for the wild strain, right? If we're comparing these two, right, with amytin, without amytin, very clearly it goes down. Right, that's pretty easy. So the effect would be to decrease the rate on the wild strain, going down. And then the experimental strain is a little trickier here, but you look at the error bars going across. They do kind of overlap, so you would say that there is no effect on the experimental strain, and that's due to this. It's hard to see, but maybe on a paper or a ruler you can see it. But they're going to overlap just a little bit, and that's enough to say that there's no difference. The average ratio, so the ratio of the average maximum elongation rate for the modified polymerase compared to the wild strain. All right, if you're going to look at figure 1, and you're looking at 12, it's the max here, and the experimental strain, if we go across, it's about halfway, which would be 2. So you're looking at a ratio of 6 to 1, 1 to 6, or 1 over 6. And so it's, keep in mind, we have, let's see how they word it here, from max modified to the wild. So you're looking at experimental versus wild. So you're looking 1 to 6. You might want to put in your answer like the ratio of 6 wild to 1 experimental, just so that it's clear for them. But again, to get that, you're looking just the max peak here, 12, down here 2, and go to the lowest number. All right. State the null hypothesis for the experiment in figure 1. Provide reasoning to justify the claim that the change in the amino acid sequence in the modified polymerase affected the shape of the active site of the enzyme. All right, so null hypothesis, I know I briefly mentioned it, but it's basically what if all, nothing, everything's kind of due to chance. There's really no difference. You kind of take what you think is going to happen in the experiment and kind of say, well, what if that doesn't affect it? That there's no significant difference between the two experimental strains, and that any difference is due to chance. That's your basic null hypothesis idea. So for this one, any difference due to the two elongation rates is due to chance. Would be one null hypothesized, or the modified RNA polymerase would not affect the elongation rate. Those would be your null hypothesis you could do. I mean, there's any number that you could pick the way, but basically, there's nothing fancy going on here. It's just minor different. Whatever's happening here is not affecting the other one, whatever difference is. Lastly, provide the reasoning to justify the claim that the change in amino acid sequence in the modified polymerase affected the shape of the active site on the enzyme. It's kind of like this because it's one of those things. Well, if that's true, then that kind of disproves the hypothesis. But this little, I'm not a big fan of the way they do that. So again, we have this. Let's zoom out here. Have that experimental strain, right, which has decreased the rate, right, which tells us that the enzyme's not working as well. So because we decrease the rate, you can say, well, that's going to be the active site because that's where all the action happens in the enzyme. So hence decreasing the rate. The last question on here was about, I have a second here, on using, we've got a cyanide, I think they had an H on there. They said, well, what macromolecules could be a precursor for this one? And I think I even asked this earlier in the year for you guys. The carbon here is in all macromolecules. H is going to be in all macromolecules. So that's not very helpful, but there's nitrogen here. Carbohydrates? Nope. Remember, carbohydrates are carbon, oxygen, and hydrogen. Lipids? Nope. Basically, same thing. A lot of CH bonds, a little bit of oxygen in there for the lipid. I'm just doing this so I can write on my board, so entertaining myself. Nucleic acids, do they have nitrogen? Yes, they do. Remember that nitrogenous base. Purines and pyrimidines, so it could be a nucleic acid. What about proteins? Protein's that amino acid, right? On board. All right, we got that. Is there nitrogen on the board? Yes, there is. So it could be an amino acid, or it could be a nucleic acid. And that's simply just looking for a protein. Simply looking for the nitrogen. It's keying on there. Phosphorus is another one that you'll see sometimes, and that's going to key in on, get too fancy with the nucleic acids. But hopefully that helps. The more we practice it, the better. Keep in mind the error bars and the axes are important. Hopefully today's work goes okay. See ya.